This video is an overview of videos that I've put into a playlist on blood vessels, as first of all, the different kinds of blood vessels and the pathway throughout the body. If anyone was interested in more details, then obviously each of these videos can go through the individual topics in greater detail. First off, when we look at the big blood vessels, whether we're looking at arteries or veins, they have comparable layers. There is an outer tunica externa, which is made of collagen. Now, that's not surprising, given that collagen is the most abundant protein in the body, and it anchors lots of things and secures structures. And so the fact that this helps to anchor the blood vessels to the surrounding connective tissue is not a surprise. The middle layer, as we'll discuss throughout this chapter, is the tunica media, which can have smooth muscle. Obviously, it doesn't have skeletal muscle. Otherwise, you would have voluntary control over whether or not your blood vessels underwent vasodilation or vasoconstriction. This is smooth muscle, which is involuntary. And when it is stimulated by either nerves or perhaps the chemical signals, then it can undergo vasoconstriction. And when it undergoes vasoconstriction, then the lumen gets smaller and less blood goes that way. Otherwise, it can undergo vasodilation, allowing more blood to flow through the wider lumen. Finally, there is an innermost lining of simple squamous epithelia, which we call endothelia. So blood vessels, whether they be arteries, veins, or even then the smaller capillaries, have this endothelial lining. It's nice and smooth. Not only does that reduce the blood pressure which is needed, but it also prevents platelets from adhering to the sides of the blood vessels, which obviously preventing platelets from initiating clots when there is no need. That is certainly very important. There are these arteries and veins with these different layers, and one can distinguish between arteries and veins. So in general, the vein has a larger lumen but a thinner wall. The artery has a narrower lumen and a thicker wall. And so then just in general, when you look under the microscope, you expect that the vein with its wide lumen and thin wall might collapse, while the artery is more likely to stay entire. And so it's easy to distinguish between arteries and veins in cross section. So while both arteries and veins have the tunica externa, tunica media, and tunica interna, that's where the endothelia is. By the time we get to the microscopic blood vessels, those other layers start to disappear. And by the time we get to capillaries, the tunica interna is the only layer of the blood vessel which is observed. So you'll see here that there is always this tunica interna, where you have simple squamous epithelia, which we call endothelia, lining blood vessels. But by this point, we don't have muscle around it and a thicker wall. So here you see red blood cells in a blood vessel, and notice how thin this cell is with its flattened nucleus. So we have simple squamous epithelia lining these. So by the time we get down to capillaries, the tunica interna is the only layer of the blood vessel which is present. Blood is on the left ventricle. It enters arteries, but there are two types of arteries. And the first type of arteries which it enters is elastic arteries. One can distinguish between the types of arteries with roles. So it has elastic, you can see here. Then there are those fibers, those elastic fibers, are often in labs, maybe lasagna noodle on the other side. And so these are elastic arteries. You need elastic connected to the walls of the artery. The heart contracts. The blood is this artery stretch. And the stretching can then create elastic recoil again. So once the pressure of the artery pumps more blood here and it stretches, there's a pressure reservoir, just as when you pull a rubber band, there's force. If you were to let go, there would be some force at the release. So the pressure reservoir then helps to bring the wall of the blood vessel back on the blood in the lumen, and this helps to push it forward. As one section of this elastic artery stretches, so with the next, then the next, and then one can feel a pulse, this wave of elastic recoil which goes over these larger arteries. Elastic connective tissue is not quite as strong as muscular tissue, and so these elastic arteries, they then are at a greater risk of developing a weakening known as which could balloon outwards, forming an aneurysm. The elastic tissue gradually gives way to smooth muscle, and then as we go farther from the heart, then we have muscular arteries. And notice in the wall of this artery, we don't see those wavy purple lines of the elastic connective tissue. Instead, we just see smooth muscle. This muscle can perform vasoconstriction and vasodilation, very important, as we will see, both in the types of, in establishing blood pressure, but then also helping to direct blood flow, because as we will see, we're constantly making decisions. Do we send blood here or blood over there? We have 5 liters of blood in our bodies, but we have 8 liters of space. And so this smooth muscle, by contracting or relaxing, establishes pressure, and then also helps to direct blood flow, as we will see. So as blood leaves the left ventricle, the arteries which are closest to the heart are these elastic arteries. These give way to muscular arteries. And then we start getting into much smaller arteries known as arterioles. It is arterioles which will then branch and form capillary beds. Capillary beds drain into microscopic veins known as venules, which then form veins. Now, we have an incredible number of blood vessels in our body. Obviously, this would vary to individual based on that individual. But as estimated to be, sequels in miles of the body, now that's not possible. But many of these vessels are microscopic. As we fill in text, you can see there are so many tiny vessels that the majority of our cells are less than a millimeter away from and so when we add all these together, it's an incredible length enough around our planet 2 times in the action. The very small arteries are called arterioles. The very small veins are called veins. So from the elastic arteries, blood flows into muscular arteries, into arterioles. In between the arterioles and venules are what are called capillaries. Now, while many people, if they were asked what's the most important part of the circulatory system, they might say the heart. I would argue that it's the capillary. The heart exudes blood here. So that it's deoxygenated blood here. So this capillary is capillaries now. As throughout the all we could go through this tube as it into the venule, and in this great tube, we could go to arterial at one thoroughfare channel at another end. That isn't the goal. The goal is not to race back to the heart as quickly as we can, but rather to have these blood pathways branch out so that all of the cells can be near a blood vessel and get their oxygen, their glucose, etc. that way. Now, by this point, the capillaries have lost that tunica media, which has smooth muscle in it, but there is a loop of smooth muscle known as a precapillary sphincter. This precapillary sphincter is at the beginning of this branching capillary bed. When these muscle cells open, then this the blood can then go from the arterial and then go into all of these branches so that the tissue will then get oxygen and glucose, etc. If these smooth muscle cells close, these precapillary sphincters close, then these branching vessels of the capillary bed are no longer an option. And so now blood does then run straight through. Now, we are constantly making decisions on where blood goes. As I had said, your body has 5 liters of blood, but 8 liters of space. And so our capillary beds are not full of blood all the time. And then we're making decisions on, do these cells need oxygen? Have we just eaten a meal and therefore I need to send more blood to the small intestine, et cetera. Here's another depiction of that. Blood is going into this capillary, but only because the precapillary sphincter is open, it is dilated. If then this precapillary sphincter constricts, then this would then limit blood flow. And these cells would only be getting their oxygen, their glucose, etc., when blood flows through. So capillary beds are branching structures, and these precapillary sphincters are the ones which then control blood flow to these tissues. Now, there are actually different kinds of capillary in the body, and this pops up in discussion every now and then in reading the chapters. So we're going through the center of this, and this capillary endothelial cells. Now, in some cases, there are such junctions between these things that can go back and forth. Non-polar gases capable of diffusing regularly. So oxygen can go out, carbon dioxide, but nothing else. And so parts of the body where we have limited transport. So for example, we have a blood-brain barrier, a blood-testis barrier. So there are parts of the body where we are trying to discourage free movement. Most allow a little movement than this, and would then be into small spaces in between these endothelials. And there you could actually have water leaving, and this water would then be things that dissolve in water, glucose, salts, amino acids, etc. So most of the capillaries of the body are continuous capillaries, and those are the things that they allow to pass. There are a couple of places in the body where, in addition to intercellular clefts, there are pores known as fenestrations. And fenestrated capillaries allow a little bit more back and forth than more typical continuous ones. And so there are parts of the body where, perhaps like spinal fluid in the is being generated, or in small intestine where we are secreting and working on more demands may be being produced. So there are a couple of parts of the body where there's a greater degree of transport for a reason. One of the key places is in the kidneys. In the kidneys, we are trying to have extra amounts of fluid leave so that urea can be filtered from the blood. And so the capillaries of kidneys are known as fenestrated capillaries because of these pores known as fenestrations. Finally, there are places where very large items need to enter or leave. So for example, in the liver makes blood proteins. Proteins are very large, but then proteins need to be able to enter or leave the blood. There are sites where white blood cells will perform phagocytosis of old, worn out red blood cells. And so in addition to smaller molecules like glucose and amino acids, proteins and perhaps even cells need to be able to enter or leave. Certainly in the bone marrow produces types of blood cells which then enter circulation. And so the capillaries, which are the most porous, which have very wide spaces between neighboring cells, are known as sinusoid capillaries. And places such as the liver, the spleen, the bone marrow, and the adrenal medulla have these types of capillaries. And so blood will travel from elastic arteries into muscular arteries, into microscopic arterioles, into capillaries. Capillaries drain into microscopic veins known as venules, which then form veins. Now, as you can see on this model, another way that one can identify a vein is not just the thin wall and the wide lumen, but then also the presence of valves. Just as there are valves in the heart, which stop blood from going in the wrong direction, there are valves in veins which stop blood from going in the wrong direction. Just as in the heart, when blood is trying to go in the correct direction, the appropriate direction, the valves open to allow this blood to pass. But if blood were to try to go in the wrong direction, for example, if the blood were being pulled by gravity back down into the foot as opposed to being pumped back into the heart, then that would be inappropriate. And so not only do veins have valves to stop blood from going the wrong way, but then also there is skeletal muscle around veins, and it is contractions here, which will then provide the pressure which push blood onward. If you were to ask, how is blood sent to, say, the feet? That's easy. The heart squeezes very hard and establishes blood pressure. And this blood pressure then pushes blood to the feet. But by the time blood returns into the veins, the blood pressure is close to zero. So how does the blood come back? Well, this is where we need more muscle contractions, but instead of cardiac muscle of the heart, it is skeletal muscle around the veins. So for example, this is why it's important that one would not want to stand at attention so rigidly, that you were not, that your leg muscles were contracting continually, because the way that you get blood back from your feet to your heart is that the skeletal muscles around the veins in your legs, they occasionally contract and relax. This generates the pressure. If your muscles were rigid, then blood would be pooling in your feet and not returning to your heart, and less would be sent to your brain. At some point you would feel lightheaded and then you could pass out. The reason for that is if you're lying horizontally, your legs are not fighting against gravity, and now it is easier to get blood to the heart. So because the veins have so little pressure, we use skeletal muscles to contract around the veins to help push them towards the heart. And then even we could include the breathing muscles in this, because these skeletal muscles, when I inhale, I create an area of low pressure. Not only is that drawing blood, not only is that drawing air towards the heart and lungs, but it's also now helping to draw venous blood back to the heart as well. And so skeletal muscles are around veins, but then also the skeletal muscles involved in breathing then also helps to create low pressure, which will transport venous blood back towards the heart.